Hello and welcome to this lesson for Higher Biology, which today is looking at breeding and field trials. So what you're expected to know in this one is we're trying to get you to understand the role that artificial selection plays in producing sustainable food. And in order to show that you can do that, we're looking for you to be able to give examples of characteristics that have been selected for in plants and in animals. You need to be able to state the purpose of carrying out plant field trials, and in plant field trials explain the importance of selection of treatments, replicates, and the randomisation of treatments. So to begin with, thinking about artificial selection and what that is. So over centuries, humans have shaped new plants and animals by selecting and breeding individuals with specific characteristics, and that process is referred to as artificial selection. It's not working with natural selection, we are artificially selecting and breeding specific plants with specific characteristics. So over time, this original brassica plant has been changed into 6 different types of brassica plants. So we've got cabbage, Brussels sprouts, kohlrabi, kale, broccoli and cauliflower, all being selected for depending on which particular aspect of the parental plant that it was the breeders wanted. Now, when it comes to breeding different organisms, there are obviously different priorities. So if you're breeding animals for meat, you're probably going to be looking for a relatively large size, and the result of that has been quite a significant change in the size of many farm animal breeds over the last few years. Now, when we're looking at producing different plants and animals in this manner, we're looking to select specific useful characteristics. So it might be pest or disease resistance, the ability to survive in particular environmental conditions, ones which produce higher food yields, or ones which give greater nutritional value. And it's pretty much the exact same characteristics that we tend to be looking for in animals. So we're looking for pest and disease resistance. We're looking for their ability to survive in particular environmental conditions. We're looking for animals which are going to give us a higher food yield, which are going to be larger, are going to produce more meat, or that are going to produce more of whichever foodstuff it is we take from them. So for example, you may be looking at cows producing high quantities of milk. We're also looking for a greater nutritional value, so we're looking for the best quality that we possibly can. And in particular, when this comes to meat, we might be looking for more leaner cuts of meat, so less fat available on the organism. When it comes to plant breeding, the main aim is to produce cultivars, or cultured varieties, that will have a good yield and growth conditions typical for that crop. So what that means is we're looking for any cultured variety that is going to grow well in the normal conditions. Final crop growth, though, is a result of both genetic and environmental factors. So a new plant variety might grow very well in a lab, however, we need to evaluate its particular performance using field trials. So plant field trials have been carried out in a range of environments, and that helps us to compare the performance of different cultivars or different treatments. And field trials can also be used to evaluate genetically modified or GM crops. So field trials have to be carefully and scientifically monitored to ensure that we've got accurate results obtained and there are no adverse effects on the environment. Remember, we're thinking about sustainability, we're thinking about producing enough food but in a sustainable fashion. So field trials are usually carried out in an area of land which is divided into specific plots, and each plot is given a different treatment, for example changing the herbicide concentration, or we might use different crop plots to grow different varieties of crop. And in order to make our field trials fair, we're trying to make sure we change only one thing, that we randomise where those plots are, and that we carry out an appropriate number of replicates. So when it comes to changing only one thing, we want to try and keep our size of plots the same. We want to keep planting density, so the number of plants in that plot should be the same. We want to try and control as many conditions as possible to make sure that any impact is due to the variable that we're investigating. So having created our cultivar, we might be looking to see how it manages with different concentrations of fertiliser. So we want to make sure that as far as possible, the only changed difference between one plant to another is that concentration of fertiliser. And obviously this is quite difficult to do when we're out in the field itself. Because of that, we need to make sure we have something called an appropriate number of replicates. Essentially, how many times you're going to be repeating that particular experiment. And what we mean by that is we just need to have enough plants in our plot to make sure that any differences take into account the variability from plant to plant. So we must perform several replicates of every single plot, and that allows us to reduce the variability that's going to occur. We also need to consider the randomization of our plots. If we're going to look at the plots in this picture, they all look pretty similar. We've got similar size, similar planting density, etc. However, if this is a trial of different fertiliser levels, we've been given 5 rows of different fertiliser levels. So that's good replication. We've got lots of plots given different fertiliser amounts. However, if there was a stream up at the top end of the photo that we can't see from here, the plants at the end would therefore have higher moisture levels, and that's going to affect their growth. So we have to consider how we randomise our plots. An ideal field trial design then has got a mixture of all of these things in it. So in this one, for example, if we consider we're comparing presence and absence of fertiliser, so absence is our blank ones, presence is our darker grey ones. So we can see we've got randomisation over 3 different locations. So no 2 locations are the same. We've got a good mixture of with fertiliser and without fertiliser in every single location, and they're not ordered or organised in any particular way. They are very random. We've only given it 2 treatments. We've given it with fertiliser and without fertiliser, so we're only looking at changing fertiliser concentrations. We've also given 90 replicates for every treatment, and that takes into account the difference in variability from plant to plant, and also takes into account a little bit between the variability from one location to another. So from that, you should be able to give us examples of characteristics that have been selected for in plants and in animals, such as looking at higher food yields, higher nutritional values, pest and disease resistance, and the ability to thrive in particular environmental conditions. You should be able to state the purpose of carrying out plant field trials, which is really to compare the performance of different cultivars or treatments, and also potentially to evaluate GM crops. That we need to be careful in terms of the selection of treatments, replicates and randomisation of treatments. So we're trying to make sure that we select treatments appropriately to make sure we ensure a valid comparison. We're using a good number of replicates to take into account the variability within the plant sample, and we are randomising our treatments in order to eliminate bias when it comes to measuring our treatment effects. As ever, feel free to ask any questions when we go over this in class.